assistance in all these topics and this presentation. We pray that we would all sense the Holy Spirit's presence among us this evening. We pray for understanding. We pray for humility. We pray that you glorify your name. You tell us to commit our ways to you and trust in you, that you will act. Here we are, Lord, and we're committing this time to you, and we trust you. So do the work that only you can do so that you alone are glorified. We pray in Jesus' name. Amen.
Amen. Good evening, folks. You know, it's a great time to be a Bible-believing Christian, as it always is, but even more so today, we have so much more knowledge, real knowledge in science, empirical knowledge from experiments that just continually confirm what Scripture has to say. So it's a lot of fun dealing with this on a continuing basis. So, valuable and vicious viruses and a vaccine. So I have this disclaimer here that what I have to say in this presentation is not the policy or viewpoint of Arizona Christian University or of Black Mountain Baptist Church. That way I keep them out of trouble. So I can handle the trouble. The position of viruses in the biological spectrum. So I want to give you an idea of where they fit into the total picture here. They can infect every type of cell. They are not thought of as living things since they are obligate intracellular parasites. In plain English means that they have to live inside of a cell. They cannot, I used the word live. They cannot reproduce other than inside a cell. They cannot multiply unless they invade a specific cell host. They have to take over the machinery of the cell. But since they can direct life processes, make changes in what goes on in the life of an individual, of a real living being, whether it be plant or animal or bacteria, they are considered to be infectious particles. So they're more than just lifeless molecules, but they are not living entities in and of themselves. So just to give you an idea of the size, so here you see representation of two different bacteria. One is a sphere. That's Streptococcus. That's just one of the bacteria. That is one that's the common cause of sore throats by bacteria. And the other one is E. coli, the one that's elongated, that is the most common cause of urinary tract infections. And you see these numbers for sizes. That's micrometer. That means 1/millionth of a meter, 1/millionth of a meter. And in relationship to that, these viruses range from 0.2 to 200ths of a micrometer. So to give you some clue in real life how to relate that to size, that a thickness of a normal piece of paper we use in our copy machines, or the thickness of an average human hair, because there's quite a range to the thickness of a human hair, but the thickness of an average human hair or a piece of copy paper is 50 times wider than the virus. Okay, so that gives you a clue as to how small these things are. They fall into four general categories of shape, whether it be spheres, long cylinders, 20-sided figures, as you see in the bottom left, or this thing that looks like it has this huge top and a bunch of legs below. And so here it is. This is an electron microscope photograph of this type of virus. And so here is a drawing of it to make it more clear. And you say, you know what? I'll bet this thing looks like a lunar lander, right? So let's take off and see what we have for you here. So we'll talk about evolutionary ideas for viral existence, how they came into being. And there are three main ideas among the evolutionists. And the first one is that the majority of them think that viruses are former bacteria that lost most everything that makes up a bacterium other than the DNA or RNA inside and the protein coat. So you see a bunch of parts there with labels. Those are just referring to the various parts of bacteria, of a general bacteria. But here would be the result of a virus coming from a bacteria. It would be two things. It would be the DNA or RNA inside and the protein coat on the outside. The one that shows the picture, let's see. Oh, wow. There. Yeah. Okay, I think I'm going to have to reorganize myself here. Am I still in the camera range? Okay, so I'll go back here a bit and say this is all right. I don't think all the animations are working either. Oh. No, there's a bunch of stuff that's not happening here. Okay. All right, so I think I'll just have to go by the screen here. So before this, there were all sorts of other structures, other layers of tissue, not tissue, but of cell membrane, 2 layers of that, and other items that were part of a bacteria. And so their theory is that the virus lost all those other structures and ended up just with a DNA or RNA inside and the protein coat on the outside called a capsid. Second main thought among the evolutionists is how viruses came into being is that somehow these very complex molecules assembled themselves and then were able to self-reproduce. And ribozyme is a contraction of the words enzyme and ribosome, so that the business of a ribonuclease, so that the business of the RNA being able to make itself reproduced. And so they imagine all of these various things that came into being by themselves and eventually became what you see here, pre-ribosome, pre-retrovirus, pre-virus. It's all speculation. There is no foundation for this in fact. And the real issue here is that they say that time plus chance equals information, but the animation's not working. So it first says time plus chance equals information. And if you give it enough time, the impossible becomes improbable, the improbable becomes possible, the possible actually happens. But in truth, time plus chance does not equal information because information has to come from an intelligent being. Information cannot arise from matter itself. Matter is used to store information, but the information has to come from intelligence, the God of the Bible. The third general category is that there are segments that move around in our DNA. And at first it was assumed that these segments moved around by random, haphazard chance processes. But with further study, we know that indeed these segments of DNA that move around are purposefully directed, and that these genes that move around turn other genes on or off in response to changing environmental conditions for that cell or for the organism. And so they say that those parts that move around became independent and became the DNA or RNA of the virus itself. Or they say that these small circular bits of DNA in bacteria became isolated with a protein coat and became a virus. But there's no foundation for any of these. What about the biblical model? Well, let's put this whole thing into context here, is that it's estimated that there are 10 million to a billion different species of bacteria. We don't know. There's so many of them out there. It would take a tremendous amount of time to be able to catalog them all by sequencing their DNA or RNA. But bacteria are the base of our life support system, both in terrestrial settings, land settings, because they're the basis of the food chain, so that small things eat the bacteria, bigger things eat those, and so on. And the same thing in the aquatic setting, in the ocean and the lakes and streams and ponds, is that, again, the bacteria in the black rectangle are the basis of the food chain, and other larger things in turn eat them. So bacteria are extremely important, and it's important to stress that the percentage of bacteria that cause disease versus those that don't and are beneficial to us and to organisms is an infinitesimally small percentage of the ones that cause problems. We have many beneficial bacteria and viruses and fungi that live inside our GI tract or on our skin that do positive things for us. Well, okay, estimated number of species for viruses that affect organisms that have more than one cell. We are multicellular organisms. 100 million, the estimated that affect bacteria, they don't know, clueless, saying it's much higher, we have no clue. And next time you go swimming in the ocean, think that they have identified about 200,000 different species of bacteria, viruses in about a teaspoon of ocean water. So when you're swimming, you're really swimming in a solution of viruses. But it's okay because those are not the kind that like our bodies. So it's fine, no problem. Well, how about the creation model viewpoint here, understanding how this all comes together? Bacteria being the base of our life support systems, the viruses are messengers taking DNA for certain traits from one organism to another organism. Like the way bees pollinate flowers, taking pollen from one flower to another flower, the same idea. So the viruses are carriers of DNA information. And a couple examples here are the genes for the ability to detoxify mercury can be transferred from one species of bacterium to another, and then they make that mercury much less toxic to us and make things better. Another example would be in the ocean with the same thing, because I think you may remember a few years ago there was somewhat of a scare with high mercury content in tuna fish, and especially tuna that were caught closer to Japan because they had dumped tremendous amounts of mercury from industry in the waters around Japan. So they started labeling that this tuna was not caught in that part of the ocean. And another type of example is a few years ago we had a really very large oil spill in the Gulf of Mexico, and you know the doomsday people saying, oh, you know, we're wiping out the planet, the Gulf is going to be destroyed. Well, not so, because we have bacteria, Alcanovorax borchomensis, that actually break down the oil and break it into very small molecules, which are then useful biologically, and they clean up the oil spills. Well, the information for this can be transferred from one bacterium to another species by these viruses, like the bees pollinating flowers. See, so that way, more species can deal with this and with different conditions that the original bacteria may not be able to do it in a particular set of conditions. If viruses were nothing but bad news, you would expect cells to be taking great pains to evolve resistance to them, and they do express resistance in some way, but they do seem awfully accommodating in others. And this is from a secular journal, secular source here at the university. So in other words, cells have specifically designed receptors to accommodate the viruses, to invite the viruses to come into the cell. Well, we have to understand that what's the role of mutations here? You know, we're told by the evolutionists that mutations are the engine of evolution, that they cause new information to happen, and that new information is selected and the organisms get better, go upward, onward, more complex. But that's not true. Because as anybody who's worked with a computer knows that garbage in, garbage out, when you have a mutation, it's loss of information or no change. And so when your computer program goes awry because there's been a mutation, a change in it, even a virus placed there by somebody, you have functioning ceasing, lost. So the evidence is accumulating that, you know, this is not what's going on with these viruses that are helpful. What they're doing is they're transferring information, keeping the information intact, and that most bacteria, most viruses are not a problem. They're not causing disease. But the ones that do have had a mutation, and so that's the business of the garbage in, garbage out, something going wrong, loss of information, loss of original function, or loss of control of a function. So an example, even though I'm giving you the example from a bacterium, the same principle holds for viruses themselves. So cholera is the example I have up here, and many of you may know that that is a disease that can kill in a matter of hours, a few hours, because what goes wrong is because of the toxin from the bacteria, the pumps in the membranes of the cells pump out the sodium and chloride, and then the water follows, and then all the person's bodily water from the circulatory system, from the space between the cells in the tissues, ends up in the gut, and then all of that water goes out the gut, and the person dies of dehydration very rapidly. So that's an example of where there has been a mutation in the original cholera bacterium, because what its chore was in the first place was to break down these crunchy shells of crustaceans. You know, when you step on a beetle, you hear it literally crunch. Well, that's the same stuff that's in the shells of these crustaceans. It's called chitin. It's the exoskeleton, the exterior skeleton. They don't have bones inside. We have endoskeletons, our bones inside. So the job of the cholera bacterium was to break this down so the ocean doesn't fill up with the shells of dead crustaceans. Shrimp, for example, crabs, lobster. So this all fits the creation, corruption, meaning the fall in the garden, then catastrophe, the flood of Noah, and then confusion, the judgment at Babel 106 years after the flood. Biblical model that things were fine until disobedience in the garden, and then judgment, and things were allowed to go awry because of our disobedience. So we have a contrast here between the evolutionary model on the left and the creation model on the right. And so their picture is from a very small unexplained gene pool. In other words, that first living cell that somehow came into existence, and they cannot explain how that happened in a natural manner, increasing due to mutations occurring and then selection of which mutations are going to be better. But even that thought doesn't hold water, because you don't, a new organism, an offspring, that isn't selected for just one mutation. It's selected for the total organism. So even that doesn't work. Until change from one kind into another kind. So yes, there are changes within a kind, like within the dog kind. Yes, you can get a change for color of a coat or something like that, but it's still a dog. It's still the same species of dog, of the dog kind. In our case, most people think of the domestic dog, or here in our desert, the coyotes. By gain of information, and that's absolutely wrong. That is anti-science, anti-genetics. But that's the mantra that's been preached for a long time now. Whereas on the other hand, we have the creation model, and that's not going to show up, but there's another drawing there showing that from a very large created gene pool, the original created kinds during creation week, then after the flood, migration and different mechanisms called programmed filling by having hybridization take place, or these genes that move around called transposons, turning genes on or off to adapt to the new conditions after the flood, or the business of the makeup of the microorganisms in our gut changing. All these things contributed to the formation of different species within each kind, so that from the original pair of, say, dogs on the ark, we now have 34 different species, or from the original pair of cats, 36 species, or from the original pair of bear kind, 8 species, or the horse kind, 8 species, etc. But change only occurring within a kind. You can't change from a dog kind to a cat kind, or a horse kind to a cow kind. The genetics simply do not allow for it, and the secular geneticists are having to acknowledge that today. And so now they're flailing around in the professional literature. What do we do? What do we do? How do we explain this? How do we come up with a new model? Because the model that's been taught for the last, combination of models for the last 150 years or so doesn't work, and they know it. And so it's a real problem for them. So the way to combine all this, it's either by loss or shuffling of information is how we get speciation occurring. not by creating whole new kinds with mutations, as evolutionists would like us to think. All right, so viral anatomy. So here are the 4 general shapes, the cylinders, the 20-sided figures, the spheres, and the lunar landers. And these are micrograph pictures from electron microscopes you see that have been colorized. And so the basic structure is inside is the DNA or the RNA, outside is a protein coat, made up of multiple molecules of these individual protein molecules that together make up the coat. Some viruses have an additional feature called an envelope, and that envelope is the cell membrane taken from the cells within which new viruses were made by using the cell's machinery to reproduce viruses. And so that envelope, this is a representation here of the plasma membrane, the outer membrane of a cell, whether it be whatever organism that was invaded by the virus. So that's the viral envelope. It actually is the membrane from the cell that was invaded. And there are enzymes in these protein coat, this capsid. Some of these, they have different functions. One of them in particular, specifically for the RNA viruses, is that they are called reverse transcriptions. They're retroviruses. Reverse transcriptase is the enzyme that converts the RNA back into DNA so that that DNA can be incorporated into the cell's machinery and then be used to make the viral RNA. That's one type of category of enzymes that the viruses have. Polymerases are another one, and these are ones that are actually involved in the process of reproducing RNA or DNA. And we have these too in our cells. All the organisms have polymerases. So these retroviruses that have that reverse transcriptase then to convert the RNA into DNA, so the cell uses it to then make the viral RNA. So this would be the representation of the virus, and then this would be the retrovirus. It won't show. It was a cartoon of a 60s hippie, retrovirus. All right, so other functions of these viral proteins involve the ability to attach to that cell that's going to be the host cell, to dock with that cell. So there are specific proteins on specific viruses that dock with specific markers on the cell that's going to be the host cell. That's why it is very specific as to what virus will infect what kind of cell in what kind of organism. All right, then there are also the ones that take over the viral machinery, the cell machinery by the virus, so the parts can be reproduced. And then there are the ones that are called, that you may have heard a lot about, the spike proteins. These are the ones that stimulate our immune system then to make antibodies against the virus. And then there are proteins that enable the new viruses that have been made, the daughter viruses, if you use that term, to be able to detach from the cell within which they were made. So here you see that the viruses have been made, reproduced, and then need to exit. And so this neuraminidase is the name of the enzyme that allows it to clip itself off and free itself from the plasma membrane of the host cell. And so this now has the envelope of the cell that it was in around its protein coat, which is around the DNA or RNA. And this is electron micrograph that's colorized to show that exact thing happening there. All right, receptors. So we have to have these very specific receptors. There's a zillion different kinds of receptors on our cells, and they have very specific functions. This is just a list of a few of them to give you an idea of the different things that are needed for these receptors, for things to happen properly. All right? So, but in particular, now I want to bring attention to the virus receptors on cells of a plant or animal. So it's very specific for a particular virus to have a particular receptor. And so here you see one for coronavirus. That's the one that's in the news these days. And then the bottom picture here would have been showing one for adenovirus, which is the one that's one of the causes of the common cold. But it's very specific. There has to be exact match. Everything in biology is three-dimensional geometry. Everything has to fit exactly so for it to happen. So we have a host range. For example, the parvovirus only will affect the dog. There's particular virus that only affect the leaf of the papaya plant down in Mexico for those papaya plants. The swine flu virus or the bird virus or the tobacco mosaic virus, and then various viruses that affect bats, for example. Or specific tissues. So we have these receptors on different tissues so that only a particular virus, such as the rhinovirus, rhino means nose, so the rhinovirus, the one that is another one of those that causes the common cold, it affects the tissues lining our upper airway, including the upper airway in young children here is a cause of croup, for example, the parainfluenza virus. Okay, or other viruses, the norovirus that affects our gut and can cause diarrhea, or a virus that affects the cells in our liver, hepatitis. There's different types of hepatitis, in this case hepatitis C, in other words, liver inflammation. And then one here for the Zika virus. This is the one you've seen in the news in the past few years where unfortunately these children come out with these very, very, very small heads in relationship to their bodies because too many of the cells in their brain are being killed, and so the skull does not grow. So I have a feeling, okay, none of those animations are working there, but this is showing those steps where there is docking, entering, the shedding of the protein coat, and then the DNA, or in the case of the COVID virus, the RNA is converted to DNA, then the DNA is incorporated into the host cell, it's reproduced, and then from that, the proteins needed for the virus are reproduced, the DNA is reproduced, the particles self-assemble inside the cell, and once assembled, they exit the cell, and then some of them take the envelope, the plasma membrane of the cell with them. This can happen anywhere from 8 to 36 hours, and then that cell is usually dead by the time this has all happened, and then these viruses that have been reproduced go on to infect other cells until the immune system has geared up and made enough antibodies to fight these off and calm it down and stop it. And so in one cell, anywhere from 3 to 100,000 new viruses can be made. So you can see they multiply like mad. Okay, how do we respond? Well, the 4 boxed types of cells, these are the various kinds of cells in our immune system, and the 4 that have the squares around them are the ones that can actually engulf, swallow bacteria or fungi or viruses or particles of a splinter or whatever that don't belong in our body. Those are foreign bodies. And macrophage literally means big eater. That's what the name means. And so these things gobble up, as you see here in the bottom left, that the one that's called the microglial cell, and that is the immune system cell in the brain, is coming across a foreign body here that's causing trouble, and it changes its shape and actually swallows it and then digests it and breaks it down. Well, that's what these 4 types of cells do. And then they present these broken down parts inside of the virus or the bacteria or the fungus into a T cell, and then that stimulates the B cells to make memory B cells and plasma cells. The memory B cells are like a filing cabinet that keeps the information of whatever you've come in contact with that you should not want in your body, such as a virus or a fungus, or unfortunately also to pollens or foodstuffs like peanuts. And then the plasma cells, B cells convert into plasma cells, and those are the factories that make the antibodies, so that you can pour out these antibodies to fight the problem. This is what an antibody basically looks like. Of course, it's very much more complex than this, but the basic plan is that this is the constant region that never changes, that with this portion here at the bottom is what, if it's part of a cell, embeds into the membrane of the cell. And then the green part is what changes and is very specific. Now, it's not that this particular antibody changes, it's changed from one antibody to a different antibody, meaning they are made differently by the plasma cells, not that it will change itself. And so how does this happen? Oh boy. All right, this is part of your DNA, and you see numbered parts and parts that are not numbered. And the numbered parts represent what's going to end up in what's called messenger RNA, the stuff that takes the information from the nucleus into the part of the cell, the cytoplasm, where the factories are, the ribosomes that convert the information from the messenger RNA into proteins. That's called translation, translating from the alphabet of the messenger RNA to the alphabet of the 20 amino acids that we use to make proteins. And so there is selection so that certain parts called exons, the numbered parts, are what become the messenger RNA, and the other parts, introns, become segments of different kinds of RNA, about 30 different kinds of RNA for regulatory purposes. And so it's the different manners in which these are sliced out and spliced together. So you may get 1, 2, 4, 5 for one type of antibody, and you may get 1, 3, 4, 5, or maybe 1, 2, 5. So that this is how we get the variety of antibodies. And to make it a little more clear how we can make 10 billion estimated different kinds of antibodies, imagine this slot machine a lot longer with some more DNA involved, and every time you pull the lever, you get a different combination. So this is how, by this selective slicing and splicing, we can make maybe as many as 10 billion different kinds of antibodies that something in this, among these combinations, is going to match the shape of whatever is out there, whether it be a particular virus, a particular bacteria, or a fungus, something like that. Does that idea come across? Okay, so that's how we can, with a very small amount of DNA, we can make huge amounts of different kinds, huge numbers of different kinds of antibodies. And this is probably considered one of the most phenomenal parts of our entire body. It just absolutely amazes the investigators who deal with this stuff. So these are the 5 different kinds of antibodies we make. The 3 on the left are the ones that are in our blood. The 2 on the right are the ones that are, the 3 on the left are the ones that are attached to cells or in the blood, and then the IgA are the ones that are in secretions. For example, newborns get IgA-type antibodies through mother's breast milk, for example. Or IgE is the one that will trigger allergic reactions, say to peanuts or cantaloupe or shrimp, whatever. Peanuts being the most common one. And so this is showing the concept of the specificity. This particular antibody will only deal with the parts that have the same shape. This one only with the parts with that shape. This one only the parts with that shape. So we have maybe up to 10 billion of these different shapes, that one of them is going to match that thing out there that we need to deal with. It's phenomenal. So we have this army of antibodies waiting to be mobilized to deal when that threat comes to us. Okay, valuable virus and bacteria versus bacteria. So here we are in the West with the bacteriophage. Bacteriophage literally means bacteria eater, the virus that eats bacteria. So here you see the cowboy virus taming the bacterium. And this is an actual electron micrograph of these lunar landers attacking, these bacteriophages attacking a bacterium. And so it injects its DNA or RNA into the cell so that it can take over the machinery of the cell. And so here you see attachment, then entry, decoding, entry, taking over the DNA, making its own stuff, assembling by spontaneous means, and then lysing, breaking the cell and getting out and infecting new cells. That's how these viruses work. Alrighty, so is the future here now? There's some very interesting stuff going on. Since there is such specificity, I can't overemphasize the specificity, a particular bacteriophage will infect a particular bacterium. And the nice thing is, they won't want to mess with any of our cells. They won't want to mess with any of the other organisms in our body. Because we have many, many, many thousands, tens of thousands, maybe hundreds of thousands of these organisms in our gut, on our skin, in our upper respiratory tract, even on our cornea that belong there, that are doing good things for us. And they don't mess with those. So they're non-toxic for our cells, the cells of these other things, and there's no use of antibiotic in this new procedure that's under investigation, and so there's no problem of allergy to the antibiotic. So this is a very nice solution that's being worked on. So just from last July, Mayo Clinic has published this research that they're doing on using these viruses, these bacteriophages, to deal with, for example, if there's an infection in somebody who's received an artificial hip. So under the previous means of dealing with it, the surgeon has had to go back in if there's a bacterial infection in that artificial joint, open it back up, clean it out, put it back together, and put the person on antibiotics. But with this thing, the hope is that we're going to be able to use these viruses, not have to reopen the surgical wound, and just use those viruses to kill those bacteria and be done with it. That's pretty slick. Okay, what about genetic diseases, meaning specifically cancer? So downtown in Phoenix is the Translational Genomics Institute, TGen for short. It's down on 3rd Street, just north of Van Buren. And their task is to take the information that we've been learning over the last 20 years of using the specific sequence of our DNA to cure diseases that are caused by mutations in DNA, not treat them for symptoms, but to cure the disease by putting in the correct DNA, the normal DNA, into that person and replace the mutated DNA. That's amazing. I mean, that's the real cure, is fixing the DNA and not having to deal with medicines for symptoms. So that's what they do, that's their job. And so the question we then ask is, why aren't bacteria already wiped out by viruses? We said there's these uncountable number of viruses. Why haven't they killed all the bacteria? Well, the bacteria have some defense against them that God put in there, and we're able to use those mechanisms to do some phenomenal things. And those defense mechanisms are called restriction enzymes. Restriction enzymes. Restriction meaning they restrict the ability of the virus to attack the bacterium. Enzymes because they are proteins that catalyze, that make reactions happen faster. And so these restriction enzymes chop up the viral DNA or RNA so that they can't cause harm to the bacterium. And they occur in places where the DNA reads the same in both directions, and that's called a palindrome. So an example of a palindrome in English is do geese see God? Now read that from right to left. And what do you get? Do geese see God? Right? So it's the same, either direction. So that's the same thing what's going on here in the DNA. So in those locations where that kind of thing happens is where these restriction enzymes from the bacteria can chop up the viral DNA. Well, we have learned to use these restriction enzymes so that we can chop out a gene that we want to use, from a cell, a human gene, and chop that out and use it to put it into a bacterium using these restriction enzymes that came from the need for the bacteria to protect themselves against viruses. So, all of these animations. So what we do is we take, for example, a well-known example that Peggy, did I see Peggy come in? There you are, benefits from, is we take E. coli bacteria, put inside them the genes for human insulin. When I first started the practice of medicine a few thousand years ago, we were using pork and cow, bovine, porcine insulin for people because we didn't have any other source of insulin for diabetics. Well, now we use these restriction enzymes to put human genes into bacteria, and they produce huge amounts of the desired product. So that in the case of human insulin, that is produced by the bacterium, it's purified, and then the people who need it, diabetics, get actual human insulin, and we don't have to deal with the problems of using insulin from cows or pigs, because when we did that, the body would eventually develop antibodies to those because they were foreign proteins, and the doses had to keep going up and up and up and up. But now we don't have that problem, thanks to these restriction enzymes. Well, so now we can use these restriction enzymes to insert genes into viruses and use those viruses as carriers to take the genes into the right cell in a person to cure that genetic disease that they have. And this is the type of thing that TGen is working on and other institutions around the world. So that the right DNA is now there and it can make the proper receptor protein so that cell can respond appropriately to the chemical messenger coming to it, or it can make the proper products and secrete that product for whatever the body needs elsewhere or for itself. So this is how we have the use of valuable use because of the existence of viruses with the need for the restriction enzymes on the part of the bacteria. And we use viruses to carry these things into the right cells because we then manufacture the right dock for that virus to go to the right cell. Isn't that amazing? Absolutely astounding. And so here's a way we can do it to engineer, in other words, put the right DNA into the right cell. We can use the viruses to attack cancer cells to treat cancer. Well, that's research going on now. And to then help cure these diseases. Okay, just a brief mention of some vicious viruses, since we talked about the valuable ones. So look, if you had a bacterial infection, I could prescribe what you're asking for, an antibiotic, but it's clearly a very painful virus. So you see the virus attacking the person in the back. And raise your hand if you've had shingles. Let me see, anybody here had shingles? Oh, one up there. Yes. Pastor. So would you say it tickled? How would you describe it? Horrific. The pain? Awful. Okay. No more descriptors? Okay. Okay. So he starts twitching if he talks about it. Okay. It's horrendous, at least. And the problem is the pain shows up before the rash shows up. And so it's hard to diagnose in those first couple days. So the virus, which has been existing in the part of your spinal column, since you had chickenpox. And then as you get older, something happens to cause stress in your body, whether it be a physical source, mental source, whatever kind of source, infection, whatever. And then that virus is reactivated, and then it multiplies and travels down the nerve and causes this pattern of the rash and pain from the spinal column on around to the midline in the front. And it can happen on both sides and at multiple levels. It just depends on where it chooses to activate. So that's one horrendous thing. Another one is the human immunodeficiency virus causing AIDS. So the virus is entered, it's in the cell, it's multiplying, exiting, and you have new viruses, and they kill off the T cells, and then that shoots the whole immune system down. And then that person has no defense against any kind of infection. And then they get kinds of infection that any person with a decent immune system would never, ever get. The Zika virus is the one I mentioned. It's actually been around, noted since the 1930s, but only recently had made it the jump to Brazil and become a contagion down there. And so, as I showed earlier, it destroys brain cells, so if the brain's not growing, the skull won't grow, and they end up with these very small heads compared to the rest of the body, the size that it should be. And this is a permanent disability for these children. It's a very big tragedy. Or Ebola. I think you've heard of Ebola. There's been too many outbreaks of it in recent years, and this one absolutely just is totally devastating. And it affects the immune system. It affects the liver, which causes the clotting factors not to be made. And so people bleed everywhere, and their tissues all just turn to mush, including the kidneys. So Ebola. So this is a situation that originally was isolated. It was from bats. And so here you have a team in their decontamination spacesuit type suits taking a person out who has died from Ebola. But we now think we have a pretty good vaccine against it now, so there's a very good hope for that. West Nile virus is of particular interest to us because we are one of the main centers for it to occur. And it affects not only people, but also affects birds tremendously. Say what?
West Nile also impacts equines a lot.
Horses. Yes. No, that's a different. That's a different virus. That's the equine virus. That's a different one.
Sorry, my bad.
Yeah, different virus.
Yeah. We have PTSD too with West Nile.
Okay. So then we have the effect on people. It's transferred by mosquitoes. It affects mainly the brain. Now, fortunately, only a very small number of people have actually died from this, but it can make them sick enough to have coma and to have some residual neurological symptoms. But I mention this one because there's more of it here than other parts of the country, here and in Southern California. So he says, we're pretty sure it's West Nile virus. And then Burkitt's lymphoma, this is an interesting one. It's a solid tumor that characteristically affects the jaw. Very different, interesting pattern. And it involves the lymphocytes, like I mentioned, T cells and B cells, those are lymphocytes in our immune system. And what happens is it causes this characteristic break in these two chromosomes, number 8 and 14, and the parts that break off swap. So part of 8 is on 14, part of 14 is on 8. And because of that, where those breaks occur, it causes loss of control of production of these lymphocytes. And so this pattern happens, Burkitt's lymphoma. Well, there's a lot of stuff that goes on that's not happy, not good. But we have this reassurance. God will wipe away every tear from their eyes. There shall be no more death, nor sorrow, nor crying. There shall be no more pain, for the former things have passed away. So that is the confidence we can have in the future, when things will be made right. They will be made perfect in the future as they were in the past, before the fall. Okay, so now let's get into specifics about COVID. All right. Gesundheit, the uncommon cold here. So here you see snakes emerging from his nose. All right. So again, that basic viral structure, the protein coat around the DNA or RNA. And viruses can be categorized as whether they have 2 strands, 1 strand, whether they're DNA or RNA. All right, and the coronavirus is a single strand RNA virus. So that's its category there. So it has to have those enzymes that convert it, once it gets into the cell, back into DNA, so then it can be multiplied and become RNA again and go on to do its stuff. All right, so these are the types of symptoms, and these are really no different than the flu, the ordinary flu. So it's hard sometimes to differentiate whether a person has flu or whether they have COVID-19. The one thing that makes it more distinctive is the loss of taste or smell. That's one thing the flu doesn't do. But not everybody who gets COVID-19 has the loss of taste or smell. So, and these symptoms are highly variable. in terms of which ones a person gets and how severe they are. I've had quite a few students at the university who have gotten it, especially the ones on sports teams where they've been in close contact. And most of them are done with it in 3 days. You know, they have a couple hours of fever, mostly feel tired, wiped out. Sometimes the stuffiness, and then they're over it. A few of them have had the business with the taste and smell. I have one student, it took them a full week to feel back to strength again, but they're young and they're healthy. But it's highly variable. All right, Dr. Genevieve Briand is the Assistant Program Director at Johns Hopkins, dealing with applied economics, and so she took the data from the Center for Disease Control website. She took their data and took a close look at it and compiled this graph showing the percentages of total deaths per age category from February of last year till September of last year, early September. So as you see from the numbers on the right, these are the age categories, 85 plus, and then each decade below that until you get to these very young people down here. And then the percentage of the total deaths, so 30% in those ones, 85 and older, and then with each younger age category, the percentages drop dramatically. So what this is showing is that even before, starting in February, before this became a pandemic, there's basically no change in those percentages. There's this very slight variation. So as, in spite of what you've seen in the news, there has not been this huge number swelling of excess deaths that we are told happened. Okay, that's one thing. Another thing is, as usual, older people die more because they're older. I mean, this is what happens. This is part of the curse. So looking at the years before, starting in 2013, you see that there's a gradual increase in the number of people dying because the population's growing. And so even for last year, the difference is about 58,000 more than the year before, and that's not out of line with some of these other years where you see increases less or more than that. So I don't understand how these folks can say that there's this 200, 300,000 excess deaths. Okay, I don't understand how they can say that, looking at these numbers. These are the CDC numbers. Now, here's some interesting data that she took from 2014 until July of last year, and this shows each winter there's a peak of deaths. Well, that happens every year. Every year more people die in the wintertime. There's more flu, heart disease is exacerbated with the cold weather, and pneumonia happens more often. I mean, more people die in the winter every year. And then each line is due to a different cause. And the top three causes are the most common causes of death: chronic respiratory disease, pneumonia, coronary artery disease, and cancer. Those are the four top killers. So every year they're the four top killers until last year's data. And so you see that pale blue line, it's way down here now. And there's a break in this scale showing that there's higher numbers here than with this graph here. So it turns out that the numbers attributed to COVID, the increased numbers attributed to COVID, are the exact same numbers as the supposed decrease in pneumonia, chronic respiratory disease, heart disease, and cancer. So does that mean that those four all of a sudden stopped happening? No, it means there's miscoding. Now, just because someone is positive for the virus doesn't mean they died from the virus or that the virus caused their death. And several of my former students who are now either in training or in practice have related to me incidences where a young, healthy fellow in his 20s was positive for COVID and died from his motorcycle accident, but was coded as a COVID death. Okay? I think one of the reasons for this is the fact that more money was paid to the hospitals for COVID deaths, and they desperately needed it because elective surgeries were stopped and their source of income was greatly cut off. Well, I do not say this to diminish at all, one bit whatsoever, anybody who has the death of anyone, especially if someone here has lost someone to death in this last year, or whether it's attributed to COVID or not. But I'm just trying to put the actual reality in perspective here, because anytime a family member dies, it's certainly a tragedy, whether it's expected or not. It's very difficult to lose that loved one. But we have this promise also, that God will wipe away every tear from their eyes, no more death, nor sorrow, no crying. There shall be no more pain, for the former things have passed away. So we can have the confidence of this promise here, that that's the way it will be in the future. And I don't know, nobody knows, only the Father knows exactly when that's going to happen. But I get the feeling it's going to happen sooner than later. Well, what was the reaction to the dissemination of this publication that was done by Johns Hopkins? Well, after about 3 or 4 weeks, it was widely disseminated, and then it became under attack, and then they had to publish a retraction, saying retracted by the newsletter. I'll leave it up to you as to the reasons for that. Now, here is interim guidance that was published by the World Health Organization in January before the pandemic. And it says, wearing a medical mask is one of the prevention measures to limit spread of certain respiratory diseases, including COVID. However, the use of a mask alone is insufficient to provide the adequate level of protection, and other equally relevant measures should be adopted. If masks are to be used, this measure must be combined with hand hygiene, hand washing, and other protective measures to prevent human-to-human transmission. World Health Organization has developed guidance for home care and healthcare settings. Wearing medical masks when not indicated may cause unnecessary costs, procurement burden, and create a false sense of security that can lead to neglecting other essential measures such as hand washing or distancing. Distancing is important. Okay, this is what they said in January of 2020. Well, the reason they say that is because if a virus, a person who's infected has the virus, and if that virus is a part of a blob of mucus that they're coughing up, then the mask will stop it. But if that virus is not attached to a blob of mucus, it will shoot right through that fence like a BB. Those animations are not functioning. So the problem is that these are airborne. The free viruses that are not attached to mucus are airborne, and they float in the air for a long time. They have a long hang time. So that's the point I'm trying to make with the football here that's kicked up in the air, hanging up there as long as, they try to have it hang as long as possible so that your other teammates can run down the field to tackle or stop the guy from running. And so these viruses, especially in an indoor setting, do stay suspended in the air for a longer period of time. Now, what do masks actually do? So here you see some work that was done in simulating coughing, and you see that stuff gets around the masks. And the homemade masks, as you'll see on the coming up chart, are not particularly effective. The ones that are most effective are the ones that are cotton flannel, double-layered cotton. Of the synthetic fabrics, 100% polyester is the best of the synthetics. But you can make a really good one by going to Home Depot, getting a shop vac filter, HEPA filter specifically, and then make your mask, okay? And the business of having it fit well to the nose is helpful so that less stuff escapes around. The best ones are the ones that are the medical grade ones that are made that N95 means it's guaranteed to filter out at least 95% of the airborne particles. And actually, they're made better than that. They actually filter at least 97 or more percent. And then the other one, Department of Energy standard, 99.97%. of those with a diameter of equal to 300 nanometers. Now, but remember though, the coronavirus is less than 300, so some of those will still get through because they're smaller yet. But these are the best two types of masks if you want to be serious about your mask. Okay, some folks use face shields. It's much easier to feel comfortable on your face with them. They gave them to us at school, and I tried using it, but all it does is make me rebreathe my hot air. And so here's what happens with these face shields, is that as the person coughs or sneezes, the stuff goes down and then across, and then even up again. So these are not helpful either. So here is how it works. 0% help is the face shield. A minimal amount of help with the masks. The ones for medical grade are almost at 100%. So the masks just don't do a whole lot, but for the small amount that they do, if the person feels safer with it, go ahead and wear it. Hand washing is important because these viruses can remain on objects, doorknobs, money, hard surfaces for varying periods of time, for a few hours, maybe even 5 hours or more. Some surfaces may be longer than that. And so when the virus travel agent asks the virus, is the doorknob okay? Yeah, that brings home the point it is important to do hand washing to not get the viruses from your hands onto yourself. Although the professional literature says they're not really sure how much it actually contributes to actually getting infected, but washing the hands is easy and safe and simple to do. Distancing is a very actually an important thing. If you're within 3 feet of the person, then you're really in the danger zone. If you're 6 to 12 feet, then you're much less likely to have a problem. And this graph is about the number of people you come into close contact with. So if in the course of 5 days, one person can actually probably infect about 2.5 people on the average. And at that same rate of contact, in 30 days, that'll end up multiplying to over 400 people. If you cut your contact with the number of people in half, then that cuts it down to 15 people over a month. Or if you cut it in half again, then you get down to only about 2.5 people in the course of a month. So keeping distance is very, very important. That's the most useful of all these things. One great thing about living here is we have lots of sunshine, and the ultraviolet light in the sunshine destroys the virus. So go out and get some sun. Get some sun. Take off long sleeves and get some sun. Also, you'll make more vitamin D. Okay, there's been misuse of the terms, mixing of terms in the press. So I just wanted to point these out for you. Isolation is keeping apart the person who's actually infected. In the hospital and emergency department, for example, we have isolation rooms in which we use negative air pressure. In other words, the air is sucked into the room from outside of the room and has a special vent to take that air out where it's dealt with, so that anybody who's infectious in that room, it doesn't go out of that room into the neighboring areas. Quarantine, this is confining people who have been exposed to those who have the disease but don't yet have the disease themselves. So in other words, you keep them at bay until it's known whether they have the disease or not. But it's not the same as isolation because they don't have the disease until they become positive. Lockdown is this government-mandated mass shutdown. All right, mass quarantine, whether people are healthy or not. And then social distancing is more of the voluntary nature where you're keeping your distance to minimize your chance of becoming infected. I'd just like to give you a little bit of history on the origin of quarantine. This is the town of Dubrovnik in Croatia on the Dalmatian coast. And back in the times of the Black Plague, the bubonic plague in Europe, in which 1/3 of all people in Europe died from the bacterium Yersinia pestis, transmitted by rats, they kept people out of the walled city, and at a safe distance from that, they had these stations where they provided food and water and kept people out of the city waiting to come in for 40 days. And if after 40 days they were still healthy, they were admitted to the city. So those of you who speak Spanish are familiar that 40 is 40, hence quarantine, 40 days. Well, again, before the pandemic, in September of 19, Johns Hopkins published this: quarantine measures will be least effective for pathogens, that means things that cause disease, that are highly transmissible, have short incubation periods, and spread through true airborne mechanisms. In other words, hang in the air. As opposed to droplets, that was meaning the ones that are stuck on mucus. As with travel restrictions, quarantine appears to delay the introduction of highly transmissible diseases, but not prevent their spread entirely. And the world, another quote here from January before the pandemic: large-scale quarantine will be ineffective and could have big negative consequences. Then in April, same fellow: lockdowns in the US should not be lifted without declines in new cases, widespread testing, and the use of non-medical masks. Something happened in between. Total change of direction. I'll let you figure it out. Well, what can you do to prevent getting sick if you're exposed, if you're taking these measures all along? And that is, make sure that your tank is full of vitamin C, vitamin D, and zinc. There are various protocols like this that have variations. Some of them say vitamin K, melatonin is here. Melatonin is the hormone made by the pineal gland in the middle of your brain that makes you sleepy. That's why after you eat turkey at Thanksgiving, you make lots more melatonin from the tyrosine in the turkey, and you get drowsy. But these things are, this is one of the protocols suggested for being able to ward off the virus. And there are others published as well. At home, if you do get it, sleep, 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 drink, 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 drink water. Because with any infection, you need more water. And good hydration just makes everything better all the way around. And don't expose yourself to other people. Watch yourself how sick you're getting. If you get much worse, then you seek care by calling your physician, not just showing up at the office, so they can be prepared. Things such as acetaminophen, which is known to you as Tylenol, are good for the achiness. Sometimes the achiness is horrendous, really, really bad. One of our fellows here at church, one of our deacons, is just totally achy beyond belief, and helps with the fever as well. And he says, I just can't help you with antibiotics. Well, actually, some do help to a degree. Those are for bacteria, but now we have several antiviral agents that really help. So Zithromax is an antibiotic. It's been used against the malarial organism, which is not a bacterium. It's a protista, protozoa. But it does have a good an effect, an antiviral effect. And then ivermectin is another one that is being touted as well. Now, there's lots of debate about how effective they are along with the hydroxychloroquine. And I'll just say that the root of the debate could possibly due to the fact that these are well-established, long-available drugs that are very, very inexpensive as opposed to new ones that are very expensive. So that's all part of the picture as well. And remdesivir is one of those that is expensive. So that one's given IV, the other was oral. Steroids are good only if the person is sick enough to need oxygen or be on a ventilator. Otherwise, not to use steroids. So here's a mention of hydroxychloroquine as well. I say mixed results because it depends on whose literature you read. Convalescent plasma, now this is great stuff because this is where antibodies have been taken from people who've gotten the disease and can be given to people who are being treated for the disease and get better much faster. And this was one of the things that was done for President Trump when he had the virus, was he was given convalescent plasma along with some of these other things. And then this last one here, for rheumatoid arthritis, baricitinib is an anti-inflammatory agent, and inflammation is definitely a big part of the picture that this virus causes. So these things that can calm down inflammation are very helpful. This is an animation that shows that research going on that a small molecule can be used to actually change the shape of the virus, and then it cannot dock with the cells and prevent infection of the cells. So that's a piece of work that's going on. And then there's a very interesting twist to this where at about some 70 universities, including the U of A in Tucson, I don't know about ASU, whether they did this, were sampling the sewage from a particular building to see if there was any virus in that sewage from that building. And then if so, then they would screen everybody in that dorm, if they lived or worked in that dorm, to see if anybody was positive for the virus. And at the bottom example, you can see here that 311 people were screened, and 2 of them were found to be positive but asymptomatic, no symptoms. So that's pretty interesting. Of course, again, these are young, healthy people. Or in North Dakota, all these different cities decided to check out their sewage and see what was showing up. Then people get into the questions of government involvement in big ways. What about the testing? Well, there's 2 basic categories for the testing, and that is testing for the virus itself or testing that you have made antibodies to the virus. All right? So just out of curiosity, how many have been tested here so far? Okay, a good half of us here. Okay, so you've had the swab up the nose. Isn't that fun? All right. So this particular one is looking for the protein, the antigen. All right, this is looking for the virus itself, saying, do you have the virus at this time? All right. The other one is then seeing if at some point in time, prior to the time of the testing, you have made antibodies. It doesn't tell you when you made the antibodies to the virus. What about the vaccine? Okay, there's a lot of discussion about that. It's very interesting. It's a new technology, and in the past, when vaccines have been made to viruses, they've taken the entire virus, and even though viruses are not living things, they call it live attenuated virus, means a weakened virus. And so on occasion, when people received those vaccines, because the virus was intact, some people, a few number of people, did get the disease for which the vaccine was meant to prevent. But this one is different. They just take a very small segment of the RNA from the virus, from the spike protein, and then your body will then deal with that small segment of RNA virus. Okay? So one of them, it says you take the surface protein, the spike protein, and the other, you take a small part of the virus RNA. So one of them is the protein, the other is the RNA. So in the one, you produce, from that RNA, you produce that protein, that spike protein. Your system makes the spike protein, but you don't have the RNA for the entire virus, just for one segment of that protein. So there's not the danger of getting the disease from the vaccine. Not the danger of getting the disease from the vaccine. All right? So then your immune system makes antibodies to the spike protein, and then whether you've made it or it's been given to you, and then you make the antibodies. So the side effects from the vaccine are these, and they're the same as flu or the disease itself, often. Not so much the loss of smell or taste, but these kinds of symptoms can happen. And you may, I mean, some people don't have any, some people have a few. It's totally variable. Well, who's affected? Let's see. again, it's older folks die more. They're just because they're old, and they do not do so well in general. Young people do well, do very well. Those who have comorbid diseases don't do so well. That's why those who have heart disease, diabetes, other things have to be more cautious. So who should be vaccinated first? Okay, so the healthcare providers and the first responders, police and medics, fire folks, the elderly, those at risk with the comorbid diseases, educators, first the elementary, kindergarten through high school, and then the college level. And so as of, I have the date here, 2/13. I think that's out of date. I have it updated, I think, on another slide here. It's more than that. Where to get it? Go to this website, or just Google Maricopa County COVID vaccine, and you'll find where to sign up. I tried to sign up, but the slots are taken. It's hard to get a slot. Recently, they opened up the Cardinals Stadium to have folks being vaccinated there, as well as the Phoenix Municipal Stadium on East Van Buren. Now it's becoming available in other locations as well. Who not to vaccinate? So folks whose immune systems are not functioning well, definitely not vaccinate them. Severe allergies, there might be a risk of a severe allergic reaction. Children less than 12, you don't need to be putting foreign RNA into them, and they do well. They don't get sick. And those who are pregnant, same issue. Want to protect that little baby from being exposed. So now here it is, 61.29 million doses as of yesterday have been administered. This represents 18.33% of the US population. The general figure is you need 70% of a population to either have had the disease or be vaccinated in order to develop what's called herd immunity, so that if that percentage of the population has it, then it's not going to spread to those who don't have it. Herd immunity. From the onset of being able to study this, at the time the study was done at 8 months, which was in December, then it showed that there was good lasting effect of the immunization. Some of the complications, the business of the severe allergic reaction, that's called anaphylaxis. That's shown up in 3 people, 2 in Britain and 1 in the US. Interesting, some folks who have had cosmetic injections to make themselves look, to have stuff injected under their skin to look not so wrinkled, not the Botox stuff, but fillers. Have had some local reactions, but easily treated. So there you see, here's the inflammation there on the left-hand side, and easily treated, restored back to normal. Small number of folks have shown paralysis in the nerves of the face, but most often those recover. When I've treated patients with that kind of situation from other causes, they have done well. We'll see with time if these folks do well. As of mid-January, 55 deaths have been attributed to the vaccine, but that hasn't been verified, so don't know what the actual number is. Long-term complications, well, we need more time to find out. Not enough time has gone by to see what long-term complications may show up. So someday, just for a change, why can't we send American flu to Asia? Mitch. Okay, DNA. Yes, the question is, what's the difference between DNA and RNA? Okay, DNA is called deoxyribonucleic acid, that its full name, and it means that there are four letters, these molecules, four letters, adenosine, cytosine, thymine, and guanine, A, T, C, and G. RNA has one different letter. It's A, U, C, and G, so it has one letter different than the DNA, and it has a different, one atom difference in the sugar that is used to link these molecules, these letters together, A, C, T, G. And in us and in multicellular organisms, there are two strands of the DNA that are connected sideways, two very long strands connected sideways. RNA is a single strand, and DNA is the storage of the information in the nucleus of our cells. That's the storage, that's the permanent record of our genetic information. RNA is made from the DNA, and it's, well, there's, like I say, 30 kinds, but the kind everyone talks about is the messenger RNA, which takes that information from the nucleus, from the filing cabinet, takes it out into the cell to the factory so that that messenger RNA can be read, the blueprint can be read, and tell the factory, the ribosome, what protein to make. Okay? Is that clear? All right. Now, so we have single-stranded RNA, we have double-stranded DNA. But these viruses come, I mean, all viruses, they come in either double or single RNA or DNA. Okay, and the coronavirus is a single-strand RNA virus. That's why it's called a retrovirus, because it has to have the enzymes to convert it back into DNA, be doubled, and then take over the cellular machinery so that its RNA can be reproduced and its proteins can be made. Biology, it's fun.
Are other antiviral medications such as Zovirax?
There are question is, are there antiviral medications such as Zovirax? And yes, the answer is true, and there are several of them, and they act in different ways. Some of them are to prevent docking of the virus with the cell. Some of them to prevent those enzymes, the retroenzymes, to make it go back into DNA. Some of them are to prevent the polymerases to be able to put the new DNA or RNA together. Some of them are to prevent the ability of the components to self-assemble, and some of them are to prevent the ability of the new virus to leave the cell. And so that's why with certain viral infections, more than one drug is used to attack at multiple points at the same time. What about the iver? How does the ivermectin work? Let me think for a second, because I have not dealt with this drug myself. I know it deals very significantly with the inflammatory effect. What it maybe does more specifically with the RNA itself, I am not sure. I'd have to dig more into that. They're trying to reproduce. That's what they're trying to do. Remember, they don't live per se. They're not living things, but they want to multiply. Now, see, this is an example of where things have gone amok, things have gone wrong, because the original purpose was to bring new information to the other organism so it could do better in its situation. So this is an example of how, because of the fall of the curse, things are not the way they should be.
I have had both doses, and I'm wondering, can I still carry the virus? Pass it from one person to another?
Usually within about 10 days, you're no longer contagious. You no longer shed the virus. Okay, this one should last longer, although they don't know how much longer, because the coronavirus is more stable. It doesn't change, it doesn't mutate nearly to the rate as the flu viruses do. And the reason why you need a new flu vaccine is because the flu viruses are very unstable, they change a lot, mutate a lot, and so you need different antibodies to prevent those infections. So that's why the flu has to be on a much more frequent basis. Only time will tell how long this one will be effective before you need a booster. Again, what they're taking is only a small segment of the RNA to either for you to make antibodies to or for you to produce that one A protein in order to make antibodies to. It's only a small segment. It's not the entire virus. Yeah. Right.
Well, I've been dosed twice, and the first time, I'm asking this for a clarification, but someone actually told me who's in the medical field that if I've gotten more effects from the vaccine on my first dose, feeling the chills and all that, than I did my second dose. And someone told me that if I had it on my first dose, all the effects come to me rapidly, then I have COVID in the past. Is it true? Because I had suspicion from back in August of 2019 when I got very sick that one time.
Okay, so what you're saying is you reacted more severely to your first dose
Yeah.
than your.
Not severely, but more aggressive is the proper term. Like I really felt the chills and everything like that.
Okay, aggressively, severely, I'd say it's the same.
Tomato.
Yeah. To your first dose and the second one, so they're saying that suggests that you had already had exposure to the virus.
That is correct from what I've been told. But I wanted to hear if that's even.
I think that's logical. It makes sense to me.
Yeah, because I remember back in August of 2019, I think that's how earliest this virus has been around, but I came from the Midwest and I had colds bad in the past, but this was not what I've experienced.
Right, and see, that's part of the problem is the symptoms from this, other than the smell business, could be just the same as a regular flu virus.
But the difference was I was coughing, it was the most violent coughing I've ever had
Mm-hmm,
at that one time. If you recall me being sicker than a dog that.
mm-hmm. Yeah, that would make you think more likely possibly to be the COVID virus at that time, but the only way to know would be if you had had a test for the antibodies prior to receiving the vaccine. If that had been done, then you would know exactly. But there was no way for you to know.
Well, only God knows.
Yeah, but that would have been the way to be able to determine precisely if that was the case.
Yeah, but, you know,
Right.
2019.
Right, yeah, I mean, yeah, why go through all that unless you feel like you have to, right? Okay, I'm sorry, louder and,
Is there a vaccine that's only one dose, and is it as effective as two?
I'm only aware that people need the 2 doses, not an all-in-one.
I think what she's asking is the new one that's coming out that's only going to be one dose.
Oh, I don't know. I don't know. There's so much about this stuff, and it keeps changing by the week. There's so much.
So from an initial exposure, how long does it take to incubate? So if you were exposed, let's say on a Thursday, how long would it be before you should get tested? If you know you were exposed on a Thursday and you went out Friday and get tested,
They're saying no sooner than 3 days. No sooner than 3 days, because you need time for it to do its thing and then for you to mount a response. Yeah, or if you're going to test for the virus itself, yeah, at least 3 days. Oh, I'm sorry. The question was how long to wait for testing after you think you've been exposed, and the answer is at least 3 days.
I was just going to say that with my injections, I had absolutely no response whatsoever. I didn't have any
meaning no symptoms, not necessarily no response. Yeah, meaning you felt okay. Right. Yeah, it's highly variable, but don't say no response because that means you didn't get the benefit of the vaccine. Right. So what you mean is you had no symptoms. Okay. All right. That's good. No symptoms with response. That's what we want. Okay, please repeat that louder. Meaning that your antibodies that you make will only last for 6 months. Okay, so I think the question is, you've gotten the infection, and your own response is only good for about 6 months, and then you're not protected. Is that what you're saying? Yeah. Okay, again, the study that I showed here from Monash University said that up to the 8 months, which had been since they were able to have the study happen, that protection was still very good. All right.
Ask Joseph for me if there have been any studies relating the vaccine and breastfeeding, any passive immunity to the baby. So it says, have been any studies relating the vaccine and breastfeeding, any passive immunity to the baby.
Right. And, okay, the question is, have there been any studies showing whether if a mom who's had the infection or the vaccine has given passively antibodies through the breast milk to the baby? And I don't know of any studies, but that should be the case because those IgA antibodies that are made are the ones that do provide passive immunization to the baby through the breast milk. So I'd say it should be happening, but I don't know of the studies. Doesn't mean they're not there, it means I don't know. The answer is non-medical. Okay, the answer is non-medical. Can you read my lips?
Can you restate that disclaimer that you gave at the beginning?
I just said it's non-medical. I thought that's a pretty good way to deal with that. The question is, why make a big fuss about this and not the flu in the past? And I'll just say the answer is non-medical. Okay, thanks for a great turnout.